Hi, Assalamualaikum warahmatullahi wabarakatuh. So, see you in this chapter, Expression of Biological Informations. Here is the objective for these studies. You can see that actually we're going to look at uh, the what is the DNA, what is the replication, and also process of protein synthesis. In this one is we have the outline. We're going to look at DNA, genetic information, DNA replication. We're going to look at the Messerschmitt star experiment, protein synthesis, on the genetics, code, transcription, translation, and also the lactose operon. We have number one. Okay, we have DNA. Alright, so here we have a DNA and genetic information. Alright, DNA is a hereditary material that contains genetic information that can be passed from one generation to another. So they consist of two polynucleotide strands that made up from a nucleotide that call each other, that will form a double helix. Okay, so the strands are antiparallel. They call it antiparallel because one of these strands are made from 5 to 3 prime, another is from 3 to 5 prime. So at the center of this DNA, the content of the what we call as nitrogenous bases are made from purine and also pyrimidine. Okay, they're held together by a hydrogen bond. So basically, here is the pictures of the DNA molecule. Okay, in terms of chemical structure. Okay, and also the molecular model. Okay, next we have a number of base pair that have been in this chromosome, which is obviously the X is larger than the Y, so X chromosome contain more base pair compared to the Y. Right, so okay, we're going to look at the Griffith and Avery experiment. Okay, this experiment is being done in 1931. Alright, in 1931, okay, at that moment, the knowledge about DNA is very limited. Then they don't know about the DNA. There is no knowledge about the hereditary material at this 1931. So this Griffith do the experiment, which by using a streptococcus pneumonia, which is a bacteria that that will cause a disease, or we call it is a can cause death. So what we call is a to their studies. In this study, they use mice. So basically, streptococcus they got two strain S and strain R. S stand for the smooth, R stand for the rough. Strain S is a smooth surface, meaning that the strain S, the content of the polysaccharide and capsule that protect the cell from the immune system. Therefore, they are virulent, meaning that strain S uh, is a bad guy, which actually when they enter to a certain organism, they are being immune. Okay, from the antibodies from the organism. Okay, so obviously strain S can cause death. Okay, both are bad. Okay, so but the strain R is a lesser, or we call it a less virulent, because they do not contain the polysaccharide capsule that protect them from the immune system. So meaning that they will can cause what we call it is a mild symptom. Okay, compare to the strain S. So here, and they can be attacked by the immune system, therefore non-virulent for the strain R. Okay, so the Griffith experiment show that basically in this experiment, what we have is, okay, the mouse will be injected with the rough strain, which is non-virulent. Okay, they are healthy because the antibody or immunity from the mice protect this organism from the rough strain. Okay, so this is a smooth virulent, as I mentioned earlier. So they are virulent, so they can protect themselves from the immune system. So the mouse is there. Okay, so compared to the heat kill smooth virulent, okay, we're taking the strain S, the smooth, and then we make it is a heat it. Okay, so basically the heat kill the bacteria. Okay, so at the end the mouse is healthy or survive. Okay, so basically when we take this one and also this one to form one mixture, okay, so which is supposedly the result is a positive, which is mouse will be alive. But at the end of this experiment, okay, Griffith found that the mouse is dead, which actually caused a certain curiosity towards the Griffith, which is how the mice is dead, okay, even though the previous experiment have been done before. So they found that actually the live stream, okay, sorry, the blood sample from this experiment found that they have a live strain S, meaning that basically the strain S just now, okay, they are killed through the heat. Okay, they put the live strain R. Okay, actually something changed, okay, in this surface of the rough strain, atau we call it the strain R, into the strain S, meaning that something have been changed from the strain R into turn to the strain S. So this is a quite shocking to the Griffith. So they just say, what happened here? Okay, so at the end of this one, conclusion, okay, so they have a strain S, strain R has been transformed, okay, they transform from the something that be transferred from the that strain S to strain R. There was something like zombie, okay, so but the nature of the, we call it a transforming principle, not be identified until 1944. So Griffith cannot found what really happened, what causing that the string, that string as turn, okay, or transfer something to the, making that string R change to the string S. Okay, so the Streptococcus pneumoniae show the something passed from the dead bacteria into a nearby cell, then making that alarm to change their cell surface to making there they are changing space from the R to S. So they don't know what is that, okay, because at that moment there is no word for the DNA and so on and so on. Okay, they just call it as a transforming principle. Okay, but they don't know what is it or how it works. Okay, until their student, okay, until the student of Griffith named as Avery found that it actually is a DNA. So next one, we have the Avery experiment in 1944. So in 1944, Avery and co-workers and their team is actually found that actually the things, or we call it the transforming principle that that they're being looking for is actually the DNA. Okay, they take a mixture of dead string S and live string R. Okay, and they remove as much as possible of the capsule or the protein, and suddenly they achieve 99% of purity. Okay, but the transforming activity is still not being reduced, meaning that it's still changed, meaning that the mice still dead. Okay, so basically it's not a protein, so basically it's a DNA is actually. So when they purify the material, elements are being RDNA. So basically when they centrifuge, so the principle have been same density as a DNA. So protein and RNA digest the enzyme do not affect the principal activities. So at the end, when they digest the DNA, okay, they destroy all the transforming activities. So basically, they can conclude that the information or transformation material or DNA is not a protein, but actually is a DNA. So in this case, they're able to do this one by transforming of the rough strain into the strain okay, so this is how they do the Avery and focus. Okay, then this one, okay, it's not a RNA, it's not a protein, it's not a lipid, it's not a carbohydrate. So basically DNA is actually the transforming principle. Okay, so that carry the information that pass from that strain S to the strain R, that making the strain R change into the virulent strain. Okay, S. Alright, so we go to the Beadle and Tatum. It's not related to the Avery. It's actually Beadle and Tatum is a scientist that working in 1941 that explain on the concept of the gene. We call it the one gene one polypeptide hypothesis. So basically, in this experiment, okay, definition of the gene itself is gene is actually a shorter segment of the chromosome that responsible for the production of specific product. Okay, actually gene is control the characteristic of the organisms. So when the use of fungus, or we call it a Neurospora coraza, that being exposed to the X-ray. As we know that X-ray is actually is element of the radiation that may cause the mutation towards the fungus. So the fungus will grow and they will develop into a fruiting body that will cause a ascus that contain 8 ascospore. Okay, there will be cultured separately in a complete medium, meaning that in a complete medium here we have all the amino acid, we have all the vitamin, we have the glucose and biotin, meaning that the fungus or the fruiting body, okay, will be developed into a mycelium, which is something that fungus, okay, fungus young plants. Okay, so some mycelium is transferred to the minimum medium. They contain a glucose and amino acid, meaning a minimum only. Okay, then if the mycelium grow, it's not a mutant, but when they are not grow, meaning that they are mutant. Okay, so if the mycelium fail to grow, then this is a mutant. So the mycelium form the original culture, then it will be cultured into 20 different minimum of the 20 different minimal medium, which is each of this one will be added with one amino acid. So here if the mutant fungus, okay, they will only grow in a minimum medium, okay, which is plus with the amino acid being they cannot synthesize. So that's why they will only grow if the but certain or specific amino acid being added to the medium. So here is the uh, illustration or the diagram how the Beadle and Tatum in the concept of one gene one polypeptide being uh, they're doing this one. Okay, so basically through the X-ray they will produce 8 ascospore. This ascospore will be in a complete medium or complete medium. If they fail, okay, sorry, the complete medium, uh, all 8 ascospore will be grown here, including the mutant one. Okay, then they will transfer a little bit, or what we call a mycelium, or we call the young plants of this to the designated test tube with the same test tube. For example, this is seed 1, they will transfer to the seed 1 also with a minimum medium. Okay, so here if the ascospore or the seed is a mutant, meaning that mutant here, meaning that a certain amino acid they cannot being produced in that particular plant, so they fail to germinate, they fail to grow. So here at the minimum medium they cannot grow. So we know that actually, for example, the test tube number 4, okay, so example test tube number 4, when they put the minimum medium, okay, so you put the mycelium, they fail to grow. We know that seed number 4 is the mutant. Okay, so when they are mutant, okay, they take the sample from the complete medium. Okay, we know that this is number 4, which is a mutant number 4. They take the mycelium, a sample of young plant here. Okay, transfer into a 20 test tube, which is each of this one will be given with the specific amino acid. If they grow at this example arginine, they grow, meaning that they know that actually there's seed number 4, they fail to produce arginine. So meaning that hypothesis from a Beadle and Tatum being accepted, okay, meaning that one gene, if one particular gene at the certain chromosome have been deleted, having a mutate, okay, or they cannot, meaning that they mutant, meaning that they cannot synthesize a certain protein. So they found that through this experiment, a certain protein is actually a specific to a certain protein, okay, a certain protein here. For example, this is arginine. So as a conclusion, so X-ray will be induced a mutation of the gene. So basically, this gene will contain a production of an enzyme that involved in the amino acid, which amino acid is actually produce a hypothesis we call it a one gene one polypeptide. Polypeptide. So meaning here, polypeptide here is a protein. Alright, so we go to the number 2, DNA replication. Alright, so here, okay. Okay, we have DNA replication being proposed by the Watson and Crick. Okay, so Watson and Crick is a friends. Okay, so they're doing the model for the DNA replication, which actually this model will, they propose that actually they are contained of two strands that are capable of unwind and separate. So each of these DNA will be formed as a base, or we call it a template for the replication. Okay, we know that DNA replication is actually producing a copy of DNA. So from one single DNA to become two, so from two become four, and so on. Okay, so what is the function or what is the use of DNA replication? DNA replication being used for the process of cell division, okay, mitosis, meiosis, and so on. So every day and every second, our cell will undergo a DNA replications. Okay, so don't worry. Okay, so in this day, okay, in this way, okay, each original DNA, okay, will give two copies of identical structure. Okay, here we have a summary of DNA replication, which actually DNA replication from a double helix, they will separate by the enzyme DNA helicase. Okay, replication is using semi-conservative model, meaning that each strand will be act as a template to form a formation of brand new strand, meaning that the original one will be separate to become two. Okay, two here. Okay, so they become two, and then another or the new one will be at the other side. Okay, so meaning that the original one become a template or become a parent type. Okay, so this will be another a new, or we call it identical. Right, so here, okay, we have two strand. Okay, once they separate, okay, once DNA is separate, they have two strand. First we call it a leading strand, another one we call it a lagging strand. So this leading strand, okay, you can watch the videos. Okay, so the leading strand is actually at the RNA, and then they will have the RNA primer. Okay, DNA polymerase will add more DNA nucleotide that will join together by the exposed bases by specific base pairing. So basically, this formation is start from 5 to 3 prime. Whereas for the another one, we call it a lagging strand. The replication occurs in a short, okay, we call it a fragment of Okazaki. This fragment are short. The fragment from 3 to 5 prime, which is opposite from this leading strand. Okay, so the open area is actually 3 to 5. That we call this is a Okazaki fragment. Okay, after they finish everything, the RNA primer will be replaced by DNA, joined by the DNA polymerase. Short fragment will be sealed together by the DNA ligase. Right. So basically, after replication, two identical of the original DNA will be produced. So based on this one, we can say that this is a leading strand. Okay, this is a leading strand. So this is the lagging strand. This is what we call is a fragment. Okay, small fragment. So actually the DNA replication direction is for the leading from here to this area, but then for this one, okay, they are from here to this, sorry, from this to this one, okay, this to this one, right, the open area from this one, okay, because they only can be made from 5 to 3 prime, their DNA replication. So once everything is settled, the Okazaki fragment are not joined together, they'll be joined together by the ligase, DNA ligase. Okay, there are three possible models were being proposed for DNA replication. First, of course, the conservative, okay, the dispersive, and also the semi-conservative. We are using a semi-conservative, okay, proposed by Watson and Crick. Okay, through a semi-conservative, as I mentioned earlier, so this is a parent, okay, strand, which is the parent will act as a template. The blue one is the blue one here is the new one, okay, we call it the daughter strand. Okay, so by conservative model, okay, parent is actually serve one, okay, another one will be totally new. Okay, what about the dispersive? It's like a patch, okay, we call it a patch, meaning that every single strand they have a new and old one, they have new and also the parent type. Okay, so this is what we call as a dispersive model. All these three model is correct, but basically our humans, okay, the Watson and Crick found that actually we are using a semi-conservative because they are more reliable, okay, okay, through their experiment, through their research. So actually we are using our DNA is actually a semi-conservative model. So this one. Okay, we go to the number 4, Messerschmitt star experiment. In this experiment, the aim or the objective of the experiment is actually to ensure that the model has been proposed by the Watson and Crick, which is a semi-conservative, is correct. Okay, so they are suggest that replication, which is each daughter contain the one side of the original and alongside with the newly synthesized strand. Okay, so they make use of the E. coli bacteria, which is contain a medium of 50 nitrogen. Okay, so they label these bacteria with the 50 nitrogen, and by many generation, all the DNA of the bacteria contain 50 nitrogen. So what happened is, after bacteria have been transformed to a medium, which is a content of 14. Okay, so they got 14. Okay, so instead of 15, okay, they transfer to a medium of the 14. Okay, for one cycle, okay, they've been extract this bacteria and see their DNA. As a result, okay, so as a result, they saw that, okay, the DNA is actually is a hybrid, or they contain both strands. Okay, one strand is a 14, another strand is a 15. So actually through this experiment, okay, Messerschmitt star experiment show that semi-conservative are being applied to the bacteria also. So basically a semi-conservative are being accepted as one of the model for the DNA replications. Right, so this is also same, okay, a semi-conservative model. Okay, so all this one, this DNA replication is occur in this area. DNA replication, okay, specifically here, DNA is replicate, okay, before, okay, normally, okay, we have here after they finish, okay, so the chromatic, okay, will be transfer, okay, sorry, will be duplicate to become, okay, once they go here, they already have another sister chromatid. So this is additional of this one, which is identical. So this is we call is a DNA replication before they go for the M phase. Okay, is either mitosis or meiosis. Okay. Right, so we go to the protein synthesis. Before we go to the protein synthesis, we must know about the genetic code. Okay, so we're talking about genetic code. Genetic code is actually a language being used by the our DNA to produce a protein, a specific protein. So gene is actually a shorter segment inside our chromosome that is responsible for a certain production of the specific protein. So this protein is actually make or control the characteristic of the organism itself. Right, so genetic information in this one will be passed from one generation to another generation. So this in form of sequence of bases that will be inside in our DNA molecule. So basically, this code or the base code is actually made from a purine and pyrimidine. We have C, we have G, and plus we have T and also A. So these bases, when we arrange in a tree bases, is a triplet code, meaning that 3 base is equal to 1 codon. Okay, 1 codon is actually there will be code for the specific amino acids. So we have 64 of triplet code. Okay, code for a specific for 20 amino acid. So basically, the features or the characteristic of the genetic code is the triplet of bases that I mentioned earlier. 3 bases equal to 1 codon. Okay, they are universal, meaning that all code for all organisms in this world. Okay, so they are non-overlapping, meaning that no base a given triplet. Okay, they are actually code for different amino acid. Okay, meaning degenerate, they are one amino acid may be coded by more than one codon. Okay, this is an example of amino acid. Okay, we have the DNA and also the RNA. Okay, so what does it mean here? We have first nucleotide, okay, first bases, second bases, and third bases. So all the 64 here will be right here. Okay, inside of this 64, we have a AUG, stand for the start codon. We call this is a start. For any of the production of protein synthesis, they will be start with the AUG. Okay, so AUG is actually the start codon for any of the production of the protein. Okay, when they have start, they also have a stop. Okay, so the stop codon here is we have these three: UAA, UAG, UGA. Okay, so they do not belong or do not code for any of amino acid. That's why we call this one is a stop codon. Okay, so so others of this one, other of the codon, you don't have to remember, you don't have to memorize. Okay, the stop codon, yeah, you have to remember. Okay, so this is an example, meaning that UAA, UAG, and also UGA. Okay, there are 3 types of RNA, okay, known as mRNA, messenger RNA. Okay, stand for the or function for DNA transcription. Okay, so for the RNA, stand for ribosomal RNA, formed in the nucleus. So basically, they are associated with the protein and to form a ribosome. Okay, tRNA or transfer RNA. Okay, so actually it's a clover-shaped molecule that they have carrying a specific 20 amino acid. So they also contain the enzyme. Okay, we call it the amino acid tRNA synthase that form a amino acid tRNA complex. So basically, these seed when they are activate, okay, when they form a complex of the amino acid tRNA complex. So one of the tRNA, there is a triplet base which is known as an anticodon. So this is we call it anticodon. So when they read the mRNA, GCC, so they are opposite from the mRNA that we call it anticodon. Right. So type of amino acid carried by the tRNA is directly related to the anticodon. Okay, so this is amino acid. Okay, that related, which is carried by the tRNA, very specific to this transfer RNA. This is how during translation, how the tRNA will help in the process of protein synthesis. Okay, so anticodon here, okay, we have here anticodon. Actually, they will carry the specific amino acid, and they are in this growing of the protein. Alright, so right now we're going to show you the DNA. Okay, so alright, so we have a synthesis of protein. There are two main stages. Okay, we have transcription and translation. Transcription is actually copying the sequence of bases from DNA into the mRNA. So a copy of the secret recipe to do a certain protein. I mean, a specific genes here. So they copy into a mRNA. So transcription happen inside the nucleus. So once transcription done, the mRNA will be out from the nucleus, okay, in the cytoplasm to do a translation. So basically, transcription is the first one, followed by the translation. House cell makes a very important proteins. So they got two phase, transcript, translation, from DNA to RNA and to protein. So meaning that from DNA to RNA is a transcription, from RNA to the protein is a translation. DNA is actually from DNA to RNA is a transcription, from RNA to protein is translation. So basically, when we're talking about making a protein, is a long process. Okay, it starts from the DNA to the mRNA. Okay, so this is transcription process. Okay, DNA is a very long strand. Okay, which is they have a lot of the genes inside of the DNA. But only a small portion of this one are needed to do a certain protein, right? So that's why they have to copying, okay, and put inside the pre-mRNA. So pre-mRNA, they will do a slicing process, or we call it the RNase processing, to make a matured mRNA, or we call it the mRNA, which is full of the or matured mRNA. So the mRNA will be go out from this one, then they do the translation process to make a protein with the help from the ribosome. Okay, next one. How RNA differ from DNA? Okay, I think they already know about this one, but I just go through this one. They got sugar. In terms of sugar, they are different. RNA stand for the ribose. For DNA is a deoxyribose. Okay. RNA contain uracil. DNA contain thymine. Okay. RNA is a single. DNA is a double. Right. This is the pictures of the RNA and DNA. Okay, so we go to our transcription process. Still inside of this nuclear membrane. Okay, so this strand will be copy of the DNA into the RNA. Okay, so basically it start from the process of the transcription. RNA polymerase will be here and will be copying the. Here is the example of how DNA change into the RNA. So we have DNA from 5 to 3 prime. Okay, example we have GC, GT, ATG, the bases. So what will be the complement RNA for this DNA? So we can say that this will be, okay, always remember C will be always with the G. Alright, A will be always with the T in a DNA, but in the RNA they will be changed into U. So G with the C, okay, C with the complement for the RNA, okay, complement for the RNA, okay, is a C, G, C, T. T will be complement with the A. Now, okay, here is the critical part A. Okay, the complement with the T in the DNA, but then now with the RNA, so you are changed into the U. T with the A and G with the C. Right, so right now this is example of how copying, okay, how the DNA will be inside our RNA. So basically this RNA will be transfer into the cytoplasm outside from the nucleus to do a process, the next process. Okay, so this is already been explained. Okay, mRNA. Okay, so mRNA is actually being read by the 3 bases. Okay, each of these 3 bases is equal to 1 codon. So AUG normally is a start codon, remember? Okay, so we read from this one, 3, 3, 3. Okay, oh sorry. So each of this one, okay, if we refer to the table of the genetic code, AUG stands for the methionine, which is a start codon. Alright, GGC stands for the glycine. So amino acid for this GGC is a glycine. UCC serine, isoleucine is AUC, GGC is a glycine, and GCA is a alanine. Remember, this is the not stand for any of the amino acid. So meaning that they are stop codon. So UAA, UAG, and UGA. Okay, that will be the stop codon. Alright. So the table being given to you for the genetic code is actually the mRNA. From the you have to read from the mRNA. Right, so this example of the amino acid being held by the. I'll proceed to the transfer RNA. Still same. Okay, stand for the anticodon. So RNA processing. Okay, so normally during the process of the from the pre-mRNA into the mRNA, pre-mRNA is normally longer compared to the matured or we call mRNA, because inside of the pre-mRNA there is an intron and exon. Intron and exon. Intron and exon. So during the process of RNA processing, the enzyme of splicing will be inserted here to be intron will be pulled out. Intron. We do not need intron. So meaning that intron is a bad. Okay, we don't want intron. We want exon. Exon is a good one. Okay, so we want exon. So during the process of processing, RNA processing, intron will be pulled out. Okay, meaning and making that the intron will be together. Right? So at the end of the RNA processing, mRNA process normally are shorter compared to the pre-mRNA. So nucleus, they leave the nucleus to the cytoplasm. We don't want intron. Okay, exon is okay. Alright, this is example. Okay, how they're being splicing. Okay, because how they process of the RNA processing during this process. So they are leave with the exon only. Okay, so this is due to the intron is contain the repetitive gene. Okay, do not change the what we call the protein characters. So basically we just need an exon to do a specific protein. Okay, because intron they contain the repetitive gene, okay, which are not being used. It's actually this one being the advantage of the human have the exon and intron together is to make sure that the virus or the bacteria cannot copy human protein. So because the enzyme to splice the intron will be available for human only. So let's say if the bacteria or the virus copy our gene, so they cannot do a specific or we call it a protein that exactly like a human productions. So exactly how the human produce. So that will be what we call is like a copyright for humans. I thought this is a ribosome. Okay, ribosome function to produce a protein. Okay, so they got two sites, P and A, which is large and small subunit. Okay, So if you look at this one, so we have the mRNA will be enter or will be in this ribosome. Okay, then what happened? They will read the first codon as a AUG at the P side. Okay, so okay, obviously they got a translation, which is a 3-step initiation, elongation, and termination. So let's do the protein. So there is AUG for methionine. Alright, so methionine is a first codon, okay, first amino acid, okay, there will be a starting, we call it initiations, okay. The transfer RNA will be coming with a specific amino acid, okay, and then they will go here, followed by the second one, okay, and each of this one, okay, if you look at this one, all right, so between amino acid number 1 and amino acid 1 there is a bond, we call it a peptide bond, okay. So and then they go to the elongation process, okay. When the third amino acid come, the first amino acid will be leave from this beside, and then it will come another one, till the finish the reading all the what we have here, the mRNA. So as long the longer the mRNA, meaning that the longer the polypeptide that they will produce, meaning that the protein are more complex. Okay, so remember this protein are not functional yet because it's only what we call a single line of the amino acid. They cannot function as a protein anymore, so they have to go to the next step. So this is only, we call it a primary protein, which is only a single line of the amino acid. So then you have to go to the secondary, tertiary protein, and lastly is quaternary, where we function as a protein. Alright, so number 5, 6, and so on, so on, so on. Last will be, let's say we take it as a amino acid number 200. So the last will be belong to the stop codon. Okay, so the stop codon is do not code for any of the amino acid, so that's why they will have a termination process. Alright, at the end of the product, so you have a primary structure of the protein, so the long thread, okay, just now they produced, and this one will be bound together by the peptide bond. Okay, so this is an example. Alright, so okay. Alright, this is the answer for the question just now. Okay, UAC will be the anticodon, AUG will be the codon, and also for DNA is a TAC. So right now there will be a pictures of how the translation takes place in the cytoplasm to make a protein. Alright, so we're talking about ribosome is only one, but when poly, meaning that lots of ribosome are taking place in one mRNA to make to make a lot of protein. So basically mRNA is generally translated simultaneously by several ribosome in a cluster, that basically we call that one is a polyribosome or polysome. Okay, so this increase the rate of protein synthesis. Obviously, the benefit of the polyribosome to increase the rate of protein synthesis. Let's say the protein needed in a very short time, so they have to call a polyribosome to make it a double, triple production of the protein. Okay, thousands of the same type of the protein can be made simultaneously. Okay, and very rapid in a shorter time. Right. So next we go to the lactose operon. Okay, lactose operon is actually a cell. Okay, basically in our genes, in our chromosome, okay, they contain more than needed. Okay, at the any one moment. Okay, so for example, gene for the pepsin production is present in all cell in our body, but they're only active in our stomach, which is to produce a gastric gland. Meaning that I can say that the stomach will have the pepsin production. Okay, so because they have their own function there, which is to digest to digest of food and so on and so on. But the code for the pepsin production is actually in all cell in our body, but they to be, we call it is a turn off. Okay, so they are not turn on because there's no function. Okay, so this is we call it how the mechanism gene can be switched on. Okay, and meaning that switch on, it can be used, and how they can be switched off when they are not being used. All right, so right now we have one of the hypotheses of being produced by a Jacob and Manet on the gene control. Okay, this Jacob and Manet do in 1961. Okay, by using E. coli. Okay, this bacteria is actually absorb nutrient, especially glucose, from the gut of the mammals, in that human. Okay, and then these bacteria genes are arranged in a group because it's like operon, okay, which is a unit for the gene expression. So basically, okay, this operon consists of a few unit of gene. First, we call it a promoter. Second, operator, and the three is a structural. Okay, and then the last one is repressor or the regulator genes. So basically, when we're talking about the operon group, they consist of all these four. Okay, function of promoter is basically to site for the RNA polymerase. Operator, okay, as X control for the transcription of the structural gene, or the allow or prevent RNA from binding to the promoter. Structural is actually code for a three genes for that particular. Okay, right now we have Z, Z, Y, and A, and the last is a regulator, code for production of protein that can bind to the operator. Okay, I give you pictures on how the here is the image of the lac operon. Okay, as I mentioned earlier, okay, they have four structure. Okay, so it start with the R, regulator. So right now here is a regulator is actually producing regulator or repressor. Okay, producing a repressor. Okay, so this one will be promoter, location where the RNA polymerase will be staying here. Okay, here the yellow one here, O, operator, is actually location where repressor will bind. Okay, and here the three structural, here we have the S, structural. Okay, which is responsible, or the gene for the specific protein. Okay, right now we have a like Z, like Y, like A. Z stand for the protein, for the enzyme beta galactosidase, like Y for permase, and like A for the transethyldease. So if you want to remember, just remember here POS, POS, promoter, operator, and structural. Okay, R, repressor or regulator is right here. Okay, so next one. Okay, mechanism of lac operon is actually control the lactose from the word lactose, meaning that milk. Okay, lactose operon. How the body will produce the enzyme for the lactose. Okay, when we digest and milk, for example, because milk contains a lot of lactose. So when the lactose is absent, meaning that we do not drink any milk, okay, and under normal environment of E. coli. So E. coli is actually inside our intestine, okay, function to produce the enzyme when we digest the milk. Okay, so when lactose is absent, okay, so repressor always being produced. Okay, they are bind to the operator, and when they bind to the operator, RNA polymerase, okay, cannot being blocked, so they cannot go to the structural gene, and as a result, no lactose digesting enzyme will be produced. So right now here, when we do not drink any milk, the gene is off, meaning that enzyme that produce the for that lactose is switch off, meaning that we do not drink milk, so obviously no enzyme being produced to digest the milk. Okay, when the host or mammal drink a milk, okay, what happened? Okay, normally when the mammals, okay, the humans, when we drink a milk, okay, the body will produce the enzyme to digest that particular food. Okay, so right now the function of the E. coli inside our body, or known as the lycoperon, they will switch on when there is a milk. Okay, so as we know, milk are made from lactose. So the lactose will be a few lactose will bind to the repressor. Okay, so basically when we drink a milk, okay, there is a lactose inside our stomach. Okay, lactose will go to the intestine. So the intestine, okay, inside of the intestine there's a lycoperon. So a small molecule of the lactose stuff will bind to the repressor. Okay, and therefore they will modify the protein shape of that particular repressor. Okay, so when the shape of this repressor are being modified, they are not fit to the operator genes. Therefore, RNA will be unblocked and they are freely to run to the three structural genes that I mentioned earlier, which is the Z, Y, and A. So when they run through this one, they will do the process of translation, transcription, okay, transcription and translation to produce these three enzymes. Basically, galactoside will be synthesized, permease, and also the transacetyldease. Right, this is the lycoperon that I showed you. Okay, this is the structure itself. Okay, so this is when the gene is turned off. Okay, so this is when the genes are turned on. Okay, if you look at this one, we know that this is a repressor. Okay, during the off, switch off. Okay, so this is a repressor, always been produced. So repressor will always bind to the position of the O, which is operator. So this is the RNA polymerase. They are being blocked. They cannot run through the ZR. Alright, so what happens when you drink a milk? Okay, a small protein, we call it a allolactose, or the lactose, it will become an inducer. When they bind to the repressor change shape. Instead of M into the U. So when they change shape, they cannot bind, or they are not fit enough to the operator. So at the result, the RNA polymerase right now here, will be located here, they are free to run through this one. So as a result, they are producing the transcription, the translation, and produce these three enzymes. So at the end of this one, so when we drink a milk, okay, enzyme will be produced. So then the milk can be digested. Okay, so there will be healthy and happy tummy. Okay, we almost finished. So this one is also the same pictures. Okay, when this is a gene turned off, meaning that there's no milk, so repressor always binds to the operator. When there is when we drink a milk, okay, lactose will bind here and they cannot fit with this one, so RNA will free to run to this one, and as a result they will produce an enzyme. So this one is very good because how the genes will be controlled. Okay, so meaning that when we have something, then the enzyme will be synthesized. Okay, what is the benefit? What is important of gene regulation? You can see that every single stage in organism development, the cell need different type of protein, different type of hormone, different type of what we call uh, enzyme. Therefore, the cell need only a small part of the genetic information to produce the protein needed. I mean, for the normal development, we don't need so many of this from the DNA itself, right? We only a small portion. Number two, for energy saving, okay, for the gene regulation, meaning that by controlling the gene expression, the cell can reduce the ATP expenditure in terms of energy saving. Okay, so meaning that we just produce an enzyme that will be needed. Okay, so the person needs this enzyme in just only a small part. Okay, so that no need for the body to produce if they do not consume that particular things. Okay, so that will be what we call is a how what is the benefit of the gene regulation. Okay, that will be the last one. Okay, thank you very much. Okay, going to see you again. Okay, for the next class. Okay, dan assalamualaikum.